Good morning, everybody. It's 4 in the morning, and here I am at Fresno State, in Fresno, in the rain, working on a project. Yes, I biked in today, so it's a good thing that I attached the fenders since, again, strangely, it seems to be raining. Our research project involves moving a worm every 4 hours to collect her offspring and study their development. Since my student, Megan, is a night owl, and I'm a morning person, we decided to split up our shifts so that I take the 4 a.m., 8 a.m. moving of the worms, and she will take the afternoon, evening, and midnight shifts. One of the things that I really love about being a scientist at California State University, Fresno, is that we have such dedicated faculty and students, especially, like my student, Megan, who are interested in developing their skills and showing off how resilient and perseverant they are. So welcome to Science in California. We have to move our worms every 4 hours because an adult Caenorhabditis nematode only lives for maybe 4 or 5 days while it produces its own offspring. And these worms, once they're adults, will have about 180 to 220 offspring that develop over about 4 days. So we have to move the worms every 4 hours because they lay multiple embryos during that period of time, and we want to be able to track development. So we need to know precisely how old each offspring worm is, and so we need to know during what short time period that worm was produced, fertilized, and then deposited on the petri dishes we grow our worms on. And here's our worm now. She's not moving very quickly. She's in the edge of the bacterial lawn, and that's usually where these worms deposit their embryos when they lay them on this petri dish. So far, we only have 4 4-hour collections of offspring. Pretty soon we'll have 24, maybe 28 of these plates to study the development of the 200 or so hybrid offspring that this hermaphrodite worm will have produced by herself. So one of the things that I study in my molecular biology and genetics lab, along with my students, is how the genetic differences between two unique parents, like in humans, when combined together in their offspring, can cause the offspring to have abnormal development. And in this case, I'm particularly curious to know whether it's the contribution of mitochondrial genetics, mitochondria being the part of the cell that produces cellular energy for us to live and develop, that might be causing this developmental delay that we've seen in our worms, where some offspring will take about a day longer, 4 days or so, to develop into adulthood than their wild type or normal siblings that take about 3 days to develop into adults. And by collecting and individually analyzing the genotype or the genetics of each offspring, as well as studying their development rate, we hope to identify the region of a chromosome that is responsible for this developmental dysfunction in a subset of the offspring that this hermaphrodite worm will produce. Well, that's it for now. I've got to get back on the bike and get home so that I can get my family up and to school and come back at 8 a.m. to do this all again. Signing out from Fresno State.